Okay, we are still on erythropoiesis. We are still on erythropoiesis. And we define erythropoiesis as the process of production, growth, development, and maturation of red blood cells. Okay? And the last class, we stopped at the stages of erythropoiesis. And we said that erythropoiesis occurs in three main stages. The first stage is the size stage or size maturation. The second one is nucleus maturation, and then the third one is cytoplasmic maturation. What is red blood cell or erythrocytes? They are a non-nucleated formed element of the blood. Non-nucleated in quotes. So that means that a mature erythrocyte does not have nucleus. Okay? Now to summarize this, if you look at this slide, you would notice that as the red blood cell grows to maturity, there is a significant reduction in their sizes. The cells which were initially large becomes smaller and smaller as it grows to maturity. That is the first thing you must note. Secondly, these large cells also have large nucleus. But as the cell grows to maturity, the nucleus becomes smaller and smaller in size until it eventually disappears. The nucleus is lost at a particular stage. And when that happens, the final stage of erythrocyte production will have no nucleus. Alright, a mature erythrocyte does not have nucleus. Thirdly, there are some cytoplasmic changes. Initially, the cytoplasm of the red blood cell lacks hemoglobin. But as the cells grow to maturity, hemoglobin appears. And this as the cell grows to maturity, hemoglobin appears. And there is an increase in the quantity of hemoglobin. So you must take note of these three main changes. The the size, maturation, nucleus maturation, and cytoplasmic maturation. Now let's go deep into the stages of red cell production. Question? Yes?
What about the color? Does it also affect the...
What about
the color?
the color?
Yes, like when it's getting to maturity, does it also affect the color? Like maybe at some certain stage, it is this kind of color, at some certain kind of color.
That is also cytoplasmic changes, because when you get to the what is the color of erythrocyte? Red. Good. And what is responsible for that coloration? What? Hemoglobin. He isn't thinking, he doesn't know. Look at you thinking. Hemoglobin is responsible for the coloration. I told you that at some point the cells lack hemoglobin, but as the cells develop to maturity, hemoglobin appears, and the hemoglobin content increases. Alright? So, but you will get to know this as we proceed. Alright? So, erythropoiesis, or red blood cell production, begins from the bone marrow. Alright? The first step of erythrocyte production is the differentiation of pluripotent stem cell to colony-forming units for erythroid. Alright? Some authorities may call that multipotent hematopoietic stem cells. It's the same thing. Pluripotent stem cells will differentiate to colony-forming units, erythroid. I explained that in our last lecture. But here, we have multipotent, I hope you can see this, multipotent hematopoietic stem cells differentiating to myeloid progenitor. Look, this is also known as colony-forming units for erythroid. Alright? CMUE. Okay? So the first step is differentiation of the pluripotent stem cell to colony-forming units for erythroid. These colony-forming units, erythroid, will further differentiate to pro-erythroblast. It's on the slide. The colony-forming units, erythroid, will further differentiate to pro-erythroblast. The pro-erythroblasts are very large cells the diameter of the pro-erythroblast is 20. Okay? And the nucleus is large. we look at the diagram of pro-erythroblast, you will discover that it has a very large nucleus. The next step is the differentiation of the pro-erythroblast to basophilic erythroblast. I said the next step is the differentiation of pro-erythroblast to basophilic erythroblast. These basophilic erythroblasts are smaller in size than the pro-erythroblast. So they have a diameter of about 15. I said they are smaller in size than the pro-erythroblast with a diameter of about 15. And their nucleus are also smaller than that of the pro-erythroblast. However, the cytoplasm of basophilic erythroblast stains with blue dye. The cytoplasm of basophilic erythroblast stains with blue dye. This is followed by differentiation. It is followed by differentiation of the basophilic erythroblast to polychromatic erythroblast. So the basophilic erythroblast will further differentiate to polychromatic erythroblast. The polychromatic erythroblasts are smaller in size than the basophilic erythroblasts, and they acquire a diameter of 12. So you can see that the sizes of these cells are gradually and progressively reducing. The same thing with their nucleus. The nucleus are also reducing in size. The polychromatic erythroblast stains with red dye and a little of blue dye. I said the cytoplasm of polychromatic erythroblast stains with red dye, more of red dye and a little of blue dye. Hemoglobin appears at this stage. So at the stage of polychromatic erythroblast, hemoglobin appears in the cytoplasm, and that is why the cytoplasm of the polychromatic erythroblast stains more with red dye, and that is an indication of the appearance of hemoglobin. Is that clear? Okay. The next step of erythropoiesis is the differentiation of polychromatic erythroblast to autochromatic erythroblast. So polychromatic erythroblast will differentiate to autochromatic erythroblast. At this stage, the cell are smaller in size than the polychromatic erythroblast, and they have a diameter of 10. Autochromatic erythroblasts have a diameter of 10. The nucleus is lost at this stage. So you can see here that the nucleus is lost. So autochromatic erythroblasts have no nucleus. This is the stage where the cell lost its nucleus. And the cytoplasm of autochromatic erythroblast stains with red dye. And this is due to the increase in hemoglobin content of the cell. Remember that hemoglobin appears at the stage of polychromatic erythroblast and disappears, and increases in content at the stage of autochromatic erythroblast. Okay. This is followed by the differentiation of autochromatic erythroblast to reticulocytes. So the autochromatic erythroblast differentiates to form what is known as reticulocytes. Also known as polychromatic erythrocytes. Alright? The cells are smaller than that of autochromatic erythroblast, and they have a diameter of 8 microns. Alright? They have a diameter of 8 microns. This is 10, this is 8. Reticulocytes have no nucleus. The cytoplasm contains more hemoglobin and mesh of network. So they have networks inside the cytoplasm. You know, when you see reticulocytes, you see some reticular formation. Alright? If you study this under microscope, or you view the reticulocytes under a microscope, you will notice that the cytoplasm have some mesh of network. Alright? And that is where the names is derived from, reticulocytes. Reticular network. Reticulocytes are already mature red blood cells. So at the stage of reticulocytes, red blood cells can appear in the circulation. Okay? So they are already mature red blood cells. In fact, the normal reticulocyte count is 1%. So the normal reticulocyte count is 1%. So an increase in reticulocyte count above 1 to 2% simply means that your bone marrows are producing red blood cells at a higher rate. Whereas, when there is a decrease of reticulocyte count below 1%, it simply means inhibition of erythropoiesis. That is, your bone marrows are suppressed and red blood cells are produced at a very slower rate. Inhibition of erythropoiesis. Okay, the last stage of erythropoiesis is the differentiation of reticulocytes to erythrocytes. I said the last stage of erythropoiesis is the differentiation of reticulocytes to erythrocytes. Erythrocytes have a diameter of 7 microns. They have no nucleus and their cytoplasm stains with red dye. Due to the increase in hemoglobin contents. So, a mature red blood cell is small in size, is devoid of nucleus, and the cytoplasm contains numerous hemoglobin which can stain with red dye. I learned this thing in my primary school. Is it difficult? Is it difficult? Not difficult. It shouldn't be difficult for you because I learned this in my primary school. I knew this in my primary school. And you're already in 200 level. If you are finding it difficult, then it means you didn't go to primary school. If this is difficult for you, it simply means You didn't go to primary school. Okay, questions? Any question? No question. I think we can now proceed. So the next thing we want to talk about quickly is blood indices.